Hi. In this section we're going to be covering the cytoplasm. Cytoplasm. The objective of this section is to describe the structure and function of cytoplasm, systole, and organelle. Cytoplasm consists of all of the cellular contents between the plasma membrane and the nucleus and has two components: the systole and the organelles, the tiny structures that perform different functions inside of the cell. The systole, or intracellular fluid, is the fluid portion of the cytoplasm that surrounds the organelle and constitutes about 55% of total cell volume. Although it varies in composition and consistency from one part of the cell to another, systole is 75 to 90% water plus various dissolved, suspended components. Among these are different types of ions, glucose, amino acids, fatty acids, proteins, lipids, ATP, and waste products, some of which we have already discussed. Also present in some cells are various organic molecules that aggregate into masses for storage. These aggregations may appear and disappear at different times in the life of a cell. Examples include lipid droplets that contain triglycerides and clusters of glycogen molecules called glycogen granules. The systole is the site of many chemical reactions required for a cell's existence. For example, enzymes in systole catalyze glycolysis. Repeat. The systole is the site of many chemical reactions required for a cell's existence. For example, enzymes in systole catalyze glycolysis, a series of 10 chemical reactions that produce 2 molecules of ATP from 1 molecule of glucose. Other types of systolic reactions provide the building blocks for the maintenance of cell structures and cell growth. Organelle. As noted earlier, organelle are specialized structures within the cell that have characteristic shapes. They perform specific functions in cellular growth, maintenance, and reproduction. Despite the many chemical reactions going on in a cell at any given time, there is little interference among reactions because they are confined to different organelles. Each type of organelle has its own set of enzymes that carry out specific reactions and serves as a functional compartment for specific biochemical processes. The numbers and types of organelle vary in different cells depending on the cell's function. Although they have different functions, organelles often cooperate to maintain homeostasis. Even though the nucleus is a large organelle, it is discussed in a separate section because of its special importance in directing the life of a cell. The cytoskeleton. The cytoskeleton is a network of protein filaments that extend throughout the systole. 3 types of filamentous proteins contribute to the cytoskeleton structure, as well as the structure of other organelle. In order of their increasing diameter, these structures are microfilaments, intermediate filaments, and microtubules. Microfilaments. These are the thinnest elements of the cytoskeleton. They are composed of protein, the protein called actin, and are most prevalent in the edge of the cell. Microfilaments have 2 general functions. They help generate movement and provide mechanical support. With respect to movement, microfilaments are involved in the muscle contraction, cell division, and cell locomotion, such as occurs during the migration of embryonic cells during development, the invasion of tissues by a white blood cell to fight infection, or the migration of skin cells during wound healing. Microfilaments provide much of the mechanical support that is responsible for the basic strength and shape of cells. They anchor the cytoskeleton to integral proteins in the plasma membrane. Microfilaments also provide mechanical support for cell extensions called microvilli. Non-motile microscopic finger-like projections of the plasma membrane. Within each microvillus is a core of parallel microfilaments that support it. Because they greatly increase the surface area of the cell, microvilli are abundant on cells involved in absorption, such as the epithelial cells that line the small intestines. Centrosome. The centrosome, located near the nucleus, consists of two components, a pair of centrioles and pericentriolar material. The two centrosoles, or centrioles, I may have pronounced that wrong. Let me begin again. The centrosome, located near the nucleus, consists of two components, a pair of centrioles and pericentriole material. The two centrioles are cylindrical structures, each composed of 9 clusters of 3 microtubules, triplets, arranged in a circular pattern. The long axis of one centriole is at a right angle to the long axis of the other. Surrounding the centriole is pericentriolar material, which contains hundreds of ring-shaped complexes composed of the protein tubulin. These tubulin complexes are the organizing centers for growth of the mitotic spindle, which plays a critical role in cellular division, and for microtubule formation in non-dividing cells. During cell division, centrosomes replicate so that succeeding generations of cells have the capacity for cell division. Cilia and flagella. Microtubules are the dominant components of cilia and flagella, which are motile projections of the cell's surface. Cilia, from the word cilia, or pronounced cilia. I'm sorry, they're not from that word. They're from, the word is cilia, in the singular, cilium. In the phonetic spelling, capital S, capital I, capital L, E, A, cilia. And the reason that I went phonetic on that one is because it means eyelash, and I thought that was interesting. Cilia, eyelash, singular, cilium, are numerous short hair-like projections that extend from the surface of the cell. Each cilium contains a core of 20 microtubules surrounded by plasma membrane. The microtubules are arranged such that one pair in the center is surrounded by 9 clusters of 2 fused microtubules, or doublets. Each cilium is anchored to a basal body just below the surface of the plasma membrane. A basal body is similar in structure to a centriole and functions in initiating the assembly of cilia and flagella. A cilium displays an oar-like pattern of beating. It is relatively stiff during the power stroke, or the oar digging into the water, but more flexible during the recovery stroke, the oar moving above the water, preparing for a new stroke. The coordinated movement of many cilia on the surface of a cell cause the steady movement of fluid along the cell's surface. Many cells of the respiratory tract, for example, have hundreds of cilia that help sweep foreign particles trapped in mucus away from the lungs. In cystic fibrosis, the extremely thick mucus secretions that are produced interfere with ciliary action in the normal functions of the respiratory tract. The movement of cilia is also paralyzed by nicotine in cigarette smoke. For this reason, smokers cough often to remove foreign particles from the airway. Cells that line the uterine, fallopian tubes, also have cilia that sweep oocytes or egg cells towards the uterus, and females who smoke have an increased risk of ectopic pregnancy or outside the uterus. So in that case, it's been discovered that you would think that smoking would paralyze the cilia in the respiratory tract, because that only makes sense. In the respiratory tract, the cilia exist inside of the lungs to move debris out. Cilia. However, smoking paralyzes cilia in general. So the cilia that exist inside the uterus, inside the fallopian tubes, are also paralyzed, which is very interesting. Because once again, you would think it would be a local reaction, but it paralyzes cilia in general. Something to think about if you take up smoking. Probably don't want to do that. Little do you know that you can affect your uterus by smoking. Flagella. Flagella are similar in structure to cilia, but are typically much longer. Flagella usually move an entire cell. A flagellum generates forward motion along its axis by rapidly wiggling in a wave-like pattern. The only example of a flagellum in the human body is a sperm cell's tail, which propels the sperm towards the oocyte and the uterine tube. Ribosomes. Ribosomes are the sites of protein synthesis. The name of these tiny organelles reflect their high content of one type of ribonucleic acid, ribosomal RNA, but each one also includes more than 50 proteins. Structurally, a ribosome consists of two subunits, one about half the size of the other. The large and small subunits are made separately in the nucleolus, a spherical body inside the nucleus. Once produced, the large and small subunits exit the nucleus separately, then come together in the cytoplasm. Some ribosomes are attached to the outer surface of the nuclear membrane and to an extensively folded membrane called the endoplasmic reticulum. These ribosomes synthesize proteins destined for specific organelle, for insertion in the plasma membrane, or for export from the cell. Other ribosomes are free or unattached to other cytoplasmic structures. Free ribosomes synthesize proteins used in the cytosol. Ribosomes are also located within the mitochondria, where they synthesize mitochondrial proteins. Endoplasmic reticulum. The endoplasmic reticulum, or the ER, is a network of membranes in the form of flattened sacs or tubules. The ER extends from the nuclear envelope to which it is connected throughout the cytoplasm. The ER extends from the nuclear envelope, or the membrane around the nucleus, to which it is connected throughout the cytoplasm. The ER is so extensive that it constitutes more than half of the membranous surfaces within the cytoplasm of most cells. Functions. Cilia move fluids along a cell's surface. A flagellum moves an entire cell. Ribosome, associated with endoplasmic reticulum synthesis proteins destined for insertion into the plasma membrane or secretion from the cell. Free ribosomes synthesize proteins used in the systole. Rough ER endoplasmic reticulum synthesizes glycoproteins and phospholipids that are transferred into cellular organelle, inserted into the plasma membrane, or secreted during exocytosis. Smooth ER synthesis, fatty acids and steroids, such as estrogens and testosterone, inactivate or detoxifies drugs and other potentially harmful substances. Removes the phosphate group from glucose 6-phosphate and stores and releases calcium ions that trigger contraction in muscle cells. Cells contain two distinct forms of endoplasmic reticulum which differ in structure and function. Rough endoplasmic reticulum, or rough ER, is continuous with the nuclear membrane and usually is folded into a series of flattened sacs. The outer surface of rough endoplasmic reticulum is studded with ribosomes, the sites of protein synthesis. Proteins synthesized by ribosomes attached to rough endoplasmic reticulum enter spaces within the ER for processing and sorting. Once again, proteins synthesized by ribosomes attached to rough ER enter spaces within the ER for processing and sorting. In some cases, enzymes attach the protein to carbohydrates to form glycoproteins. In other cases, enzymes attach the proteins to phospholipids, also synthesized by rough endoplasmic reticulum. These molecules may be incorporated into the membrane of organelle, inserted into the plasma membrane, or secreted via exocytosis. Thus, rough endoplasmic reticulum produces secretory proteins, membrane proteins, and many organellar protein. Thus, rough ER produces secretory proteins, membrane proteins, and many organellar proteins. Smooth endoplasmic reticulum extends from the rough endoplasmic reticulum to form a network of membrane tubules. Unlike rough endoplasmic reticulum, smooth ER does not have ribosomes on the outer surface of its membrane. However, smooth ER contains unique enzymes that make it functionally more diverse than rough ER. Because it lacks ribosomes, smooth ER does not synthesize proteins, but it does synthesize fatty acids and steroids, such as estrogen and testosterone. In liver cells, enzymes of the smooth endoplasmic reticulum help release glucose into the bloodstream and inactivate or detoxify lipid-soluble drugs or potentially harmful substances such as alcohol, pesticides, and carcinogens, which are cancer-causing agents. In liver, kidney, and interstitial cells, a smooth endoplasmic reticulum enzyme removes the phosphate group from glucose 6-phosphate, which allows the free glucose to enter the bloodstream. In muscle cells, the calcium ion that triggers contractions are released from the sarcoplasmic reticulum, a form of smooth ER. Clinical connection. Smooth ER and drug tolerance. One of the functions of smooth ER, as noted earlier, is to detoxify certain drugs. Phenobarbital developed changes in the smooth ER in their liver cells. Let me read that again. I think I skipped something. Clinical connection. Smooth ER and drug tolerance. One of the functions of smooth ER, as noted earlier, is to detoxify certain drugs. Individuals who repeatedly take drugs, such as sedative phenobarbital, develop changes in the smooth ER in their liver cells. Prolonged administration of phenobarbital results in increased tolerance to the drug. The same dose no longer produces the same degree of sedation. With repeated exposure to the drug, the amount of smooth ER in its enzymes increases to protect the cell from its toxic effect. As the amount of smooth ER increases, higher and higher dosage of the drug are needed to achieve the original effect. The Golgi complex, or the Golgi complex, spelled Golgi, G-O-L-G-I, pronounced Golgi. Most of the proteins synthesized by ribosomes attached to rough endoplasmic reticulum are ultimately transported to other regions of the cell. The first step in the transport pathway is through an organelle called the Golgi complex. It consists of 3 to 20 cisternae. Cisternae meaning cavities, singular is cisterna. Small, flattened membranous sacs with bulging edges that resemble a stack of pita bread. The cisternae are often curved, giving the Golgi complex a cup-like shape. Most cells have several Golgi complexes, and Golgi complexes are more extensive in cells that secrete proteins, a clue to the organelle's role in the cell. Cisternae, spelled S-A-S-T-E-R-N-A-E, cisternae, at the opposite ends of a Golgi complex, differ from each other in size, shape, and enzymatic activity. The convex entry, or CIS face, is a cisterna that faces the rough ER. The concave exit, or transface, is a cisterna that faces the plasma membrane. Sacs between the entry and exit faces are called medial cisternae. Transport vesicles will be described shortly. Transport vesicles from the ER merge to form the entry face. From the entry face, the cisternae are thought to mature, in turn becoming medial and then exit cisternae. Different enzymes in the entry, medial, and exit cisternae of the Golgi complex, or Golgi complex, permit each of these areas to modify, sort, and package proteins into vesicles for transport to different destinations. The entry face receives and modifies proteins produced by the rough ER. The medial cisternae add carbohydrates to proteins to form glycoproteins and lipids to proteins to form lipoproteins. Once again, the medial cisternae add carbohydrates to proteins to form glycoproteins and lipids to proteins to form lipoproteins. The exit face modifies the molecules further and then sorts and packages them for transport to their destinations. Proteins arriving at, passing through, and exiting the Golgi complex do so through maturation of the cisternae and exchanges that occur via transfer vesicles. In steps, it goes like this. Proteins synthesized by ribosomes on the endoplasmic reticulum are surrounded by a piece of the endoplasmic reticulum membrane, which eventually buds from the membrane surface to form transport vesicles. Transport vesicles move towards the entry face of the Golgi complex. Fusion of several transport vesicles creates the entry face of the Golgi complex and releases proteins into its lumen. Proteins move from the entry face into one or more medial cisternae. Enzymes in the medial cisternae modify the proteins to form glycoproteins, glycolipids, and the lipoproteins. Transfer vesicles that bud from the edges of the cisternae move specific enzymes back towards entry face and move some partially modified proteins towards the exit face. The products of the medial cisternae move into the lumen of the exit face. Within the exit face, cisterna, the products are further modified and are sorted and packaged. Some of the processed proteins leave the exit face and are stored in secretory vesicles. These vesicles deliver the proteins to the plasma membrane, where they are discharged by exocytosis into the extracellular fluid. For example, certain pancreatic cells release the hormone insulin in this way. Other processed proteins leave the exit face in membrane vesicles that deliver their contents to plasma membrane for incorporation into the membrane. In doing so, the Golgi complex adds new segments of plasma membrane as existing segments are lost and modifies the number and distribution of membrane molecules. Finally, some processed proteins leave the exit face in transport vesicles that will carry the protein to another cellular destination. For instance, transport vesicles carry digestive enzymes to lysosomes. The structure and functions of these important organelles are discussed next.